Hi, my name is Haley Blackwell, and I will be presenting a slideshow about the evidence behind junk DNA. Does junk DNA serve functions? Topic of presentation. Is there evidence that so-called junk DNA serves significant biological functions, or is it for the most part without any function at all? Starting off with protein coding, you can see an example of how protein coding works, and I'll later explain it throughout the slideshow. Protein coding intro. DNA sequences that are transcribed into mRNA, protein coding portions of many genomes are small, and humans are found to be less than 2%. Large amounts of many genomes have been considered to have no function at all. Going deeper into protein coding, pre-mRNA is processed to form a mature mRNA molecule. This could be translated to build the protein molecule, polypeptide, encoded by the original gene. Translation machines are found in the ribosomes. Starting off with eukaryotes, the mature mRNA molecules must leave the nucleus and continue to travel to the cytoplasm, where the ribosomes are located there. With prokaryotes, ribosomes are able to attach to mRNA while it is still being transcribed. Protein coding can be found in the coding DNA sequence, also known as the CDS. This is the coding region of genes, a portion of a gene's DNA and RNA that codes for protein. Next, we have non-protein coding DNA. Starting off with a little intro, RNA that is not translated into a protein. It's also known as junk DNA by scientists, and it can serve critical roles in gene regulation. Peptides are a result of non-protein coding DNA, which are short chains of amino acids which are not considered full proteins. Some non-coding DNA regions are called introns, which introns oftentimes have enhancers in them, which are known to regulate elements. These can be found in protein coding genes and are removed before a protein is made, and they can also be found in intergenetic regions, which are found between genes. Going deeper into non-coding DNA, 98% of human genome are non-coding DNA, while the 10% of the prokaryote genome are non-coding DNA. It's a bigger trend toward greater non-protein coding DNA and more complex organisms, and this can be found in protein coding genes and are removed before a protein is made. Going deeper into the subject, selfish genetic elements, or also known as SGEs, suggested that a significant portion of non-protein coding DNA in species with large genomes may be attributable to SGEs, which these come in many forms. One of the forms are transposons, I don't know how to say it, able to move from one location to another in the genome. They leave copies of themselves in their original genetic location, and the transportation actually leads to an increase in the number of transposable elements in the host of the genome. They favor their own genetic propagation only, and often to the detriment of the rest of the genome. In this area right here, you can see the intragenetic region found between the genes, and there's another diagram right there. So, my conclusion: evidence suggests that non-protein coding DNA have no importance and are very useful when in comparison to protein coding DNA. It seems as though the evidence proves non-protein coding DNA are useless. On the other hand, evidence suggests that non-protein coding DNA has its own importance. For example, transportation leads to an increase of transposable elements in the host genome. These elements are useful because they increase genetic diversity. According from the research I've done, I personally believe the evidence proves non-protein coding DNA to be important, as non-protein coding DNA takes on many different types and forms. Lastly, I have this little diagram showing coding regions and the non-coding regions. And that wraps up my slideshow. Thank you for listening and taking your time. Have a good one.